Good evening, everyone. Thank you for joining us for the second TrEd Career Chat at AUB. The TrEd Career Chat series is the 10th initiative of our Transformative Education TrEd program at AUB. It was created to connect students with professionals who once walked these same halls, launched their careers here at AUB, and have since gone on to shape the world in remarkable ways. Our Mentoring Talk series has showcased Nobel laureates and global figures sharing powerful stories of resilience. Career Chats, on the other hand, zoom in on what happens after AUB, on the journey beyond, highlighting the bold steps taken by chemistry alumni who have built meaningful careers grounded in purpose, creativity, and impact. Thus, today we invite you to journey behind the scenes of remarkable professional paths, many of which are rooted in an institution that has endured the test of time and touched countless lives. It is here at the American University of Beirut. Tonight's session centers on a field that is often misunderstood: chemistry. To many, it's simply another subject, but to those who truly engage with it, chemistry is a mindset. It's a field that builds problem solvers, inventors, and change makers. Chemistry equips us with the skills to unlock the building blocks of life, and those who master it, like our guest today, often redefine what's possible in health, science, and technology in ways many of us never imagined. Walter White, the main character of the hit TV series Breaking Bad, once said, quote, "Chemistry is the study of matter, but I prefer to see it as the study of change," end of quote. Dr. Tarek Zeidan, our proud AUB chemistry alumnus, is one of those individuals who transform science into impact. He is an inventor, a biotech leader, and most importantly, someone deeply committed to mentorship and human development. With over 15 years of experience in the pharmaceutical and biotech industries, Dr. Zeidan has played an instrumental role in developing medications for conditions such as cancer, schizophrenia, and beyond. He is a co-inventor of LinkRx, a revolutionary platform that redefined how drugs are delivered over weeks instead of days. His platform enables long-acting therapies that have changed lives, transforming how medications are administered and improving patient outcomes globally. From his academic beginnings right here in our department at AUB, to earning his PhD at Florida State University and completing a postdoctoral fellowship at Northwestern University, Dr. Zeidan has built an exceptional and remarkable career while simultaneously holding key leadership roles at Alkermes, Lindra Therapeutics, and currently at LifeMine Therapeutics, where he continues to push the boundaries of drug discovery. Using cutting-edge genomic tools, they are discovering nature's hidden blueprint for healing compounds, a testament to how chemistry is not static, but evolving in fields like AI, genomics, and biomedicine. I am joined on this panel by Christelle Zeidan, the president of the TrEd Student Club, and Farah Al-Awar, the president of the ACS Student Chapter at AUB. So tonight, as we listen to Dr. Zeidan, let's do more than simply admiring these accomplishments. Let us draw inspiration from them and recognize how chemistry, blended with curiosity, passion, and purpose can lead to unforeseen opportunities. Please join me in welcoming someone who reminds us that chemistry is not just about equations, it's about real-world impact. Welcome home, Dr. Zeidan. The floor is yours.
Thank you, Bilal, and thanks for having me. I'm very humbled by the introduction, first being part of the long list of people who I really admire and look up for and well known. And by the introduction of my journey, it's already exhausting listening to it. But all this exhaust and challenges disappear if you like what you're doing. Enjoy what you guys choose to do in life, and all trouble will be overcome. So I would like to have this session as informal as possible. So please ask questions. Any question that can come to your mind, please feel free to ask. I'll try to answer it to my best of knowledge and help you guys on starting your career journey. So I prepared just a few slides just to walk you through my journey and my story that is still unraveling, but maybe just give you a flavor of where I started and how I ended up where I am. So I tend to talk a lot. So Bilal, stop me if you think you need more time for people to ask. So feel free to do that. I won't be offended at all. All right. So as Bilal mentioned, actually my passion and my love to chemistry started in the 8th grade. I went to Rafiq Hariri High School in Saida. And I think on the 8th grade, this is where I started kind of chemistry becomes something that, I really enjoy. And the reason, and maybe many of you experienced that, is because of a teacher. And the teacher who taught me chemistry, for some reason, loved me as a kid. She was always giving, the rest of the class kind of look at Tarek, how he's doing and how he's behaving, even though I was like a C plus, maybe B minus student. I wasn't like an A student at all. But for some reason, she liked me, and because of that, I liked probably the chemistry topic. And then I really get into it in the last year, the terminal. And this is when I started learning about organic chemistry. For some reason, organic clicked with me. I loved the material. I loved how bonds and the carbon, the oxygen, double bond, single bond, and I really understood it. And so I'm going to jump immediately into AUB, but if time allows, I'll let you know how my conversation with my family went into going into chemistry, because almost everyone in my family is going into business. We have a family business, and they wanted me to join the family business, but for some reason I really had the passion for chemistry. Anyway, I applied to AUB. I was fortunate to be accepted at AUB as chemistry major. Went through the 3 years, but this 3 years made a big difference for me, not only on my personal and personal growth, but also academic. And as all of you have heard, and maybe some of you worked, I'm pretty sure Bilal is someone who I really admire to do what he does for our late Professor Haddadin. I was fortunate enough that I took 2 classes or 3 classes with Professor Haddadin. And during my junior year, I think we still have, Bilal, 211, 212. Are these still there, these courses? Yeah. So I think it was a 212, and he was teaching us about Carl Fisher and the carbohydrates and all these things. And he just mentioned during one of the session, at that time he was a vice provost, I think. He was in more of an administration role as well. And he mentioned, hey, I'm interested, or I'm be looking for someone to work in my lab during the summer. And then he carried over with his session. So something in me just pushed me and said, I approached him and said, listen, I'm interested. And he looked at me and said, very, very, very nice, very nice. So anyway, I worked with him during the summer, doing some synthesis of small molecules and then testing them with biology department. Can't remember the professor that we were working with, but it was kind of a topical administration on the skin of the rats. And this was my first experience in lab as a professional, right? I got paid a stipend, so I was very proud that actually I made money out of something that I really like. And then there was a professor who spent 1 year at AUB as his sabbatical. His name was Professor Joseph Schlenov. And he taught me analytical chemistry, 2 courses, and then I took, I think, 1 graduate courses with him. Anyway, at the end of the year, of the junior year, he approached me and says, "What's your plan?" And I was off the bat. I didn't want to continue as medical as many of my colleagues and my classmates. They were taking chemistry as pre-med and they were preparing with medical school. I didn't see myself up to for medical. I'm not made for to be a medical doctor. I really liked research, and as you can imagine, in the late '90s, at that time, research at AUB and at in Lebanon wasn't that of interest, and I had no idea that I'm gonna travel and leave Lebanon. I was taking it day by day, or year by year. We'll see what will happen when I graduate. Anyway, he handed over the application for Florida State, and he said, "Why don't you apply?" I said, "Yeah, sure, I'll do that." And luckily, I have another friends who also applied, and we both got accepted. And we took the journey to Florida State. On the other side of the world, I had no idea what to expect. It was really this gut wrenching kind of in my stomach. Where I'm going? What am I doing? Going all these by myself, and I have no family outside Lebanon. It's just by myself. I was lucky to have friends that I know, that my friend who were we ended up roommates at the first year at Florida State. Joining Florida State, and funny, like people who says, "Oh, you're in Florida, Miami, yeah, that's good for you." I can tell you, Tallahassee, where Florida State is, and Miami is like 9 hours drive. It's very far. Tallahassee is practically in Georgia, in the north of Florida, but and it's the capital of the state. So it has nothing but the two or three universities. It's a college town. Good for grad school because you really focus. You don't have a lot of distraction through that. Anyway, I joined a group. The professor Igor Alabugan. He was from Russia, and he started his lab the same year that I started. So it was kind of me helping him building the lab. And as you can imagine, someone who's just started, because it's all new ideas, like he doesn't have an established lab, a lot of failure. But if something works, it's great. Then you can publish immediately. And so it has this, double-edged sword in it. But it was a lot of learning from him about research. During that time, I worked on things called enediones. This is a research where there are natural products extracted from, I think, ocean mollusks or species from the ocean, And these are complicated molecule, but it has a functional group that has a double bond attached to three triple bonds and called ene-diene. And these molecule, natural occurring molecule, turn out to be able to kill cells by triggering a reaction of these warheads, ene-diene, making biradicals. And these radicals can extract hydrogen from the DNA and kill the cell. But the natural occurring products do not differentiate good cells by bad cells and cancerous cells. So anyway, the research was how to make it more targeted. And so we worked on, steric effect, making it more reactive when you put something close to this function. We did this, how it to be triggered, whether it's by heat or by pH, or in some cases by light, photochemistry. This is where I got introduced to photochemistry, very, very, fascinating field, the interaction of light with matter. Anyway, we published several papers, learned a lot. The main thing that I learned from this is how to approach a problem. If we have a problem, there are different ways of approaching it. And during my grad school, the main thing, main takeaway is how to think of a problem, how to approach it, what questions need to be asked. Ask a lot of questions first, rather than jumping immediately into solving the problem. So this was a training that I took from my grad school. And as I was trying to graduate, I always wanted to do a postdoc, an additional research opportunity. And this is, personally, just because I always wanted to think, maybe I'll go back to Lebanon and if I were to do anything in Lebanon, maybe an academic career and a postdoc could be something plus on my resume. So I decided to move from one extreme, weather-wise, Florida, very humid, very hot, to Chicago, which in this case, 9 months of really, really cold weather with a lot of snow. So I moved to Northwestern University. The campus for research is North Chicago. Chicago is a very beautiful city. I really loved it. Not the weather necessarily, but during the summer, it's a really nice city. And I joined a lab, also another kind of extreme, a professor, Fred Lewis, who's was about to retire. So I'm going from a very young lab, professor lab, that I had to help build his lab, all the way to another extreme where a very established, very well-known professor who made a really significant impact during his career on organic and physical organic chemistry. But I also wanted to make a change during in my research, and at that time there are two main things that I was thinking: either material scientist or more of a medical pharma research. And the research that I worked at Northwestern was interesting at this time, and in the early 90s, there was a big hype about using DNA as molecular wires. So what does it mean? It means that the DNA, because of the shape of the DNA, where you have it as a double-stranded and kind of a ladder where you have a lot of pi stacking, can you pass a charge across the DNA, so use it as a wire, in chips, etc. So that was a lot of, and there are two main school of thought. One saying, yep, it can be used, and one, no, it cannot be used. And the reason, both groups or school of thoughts published tons about this, and there was a lot of really healthy debate in a lot of national meeting and international meeting between these two poles. That's intrigued me, and I attended one of Fred's talk a couple of years before I joined. And really, I was intrigued by his approach and his research. Anyway, so joined his lab, worked on synthesizing kind of short polymers, where a lot of by stacking, and we showed that it doesn't matter how long the difference between the donor and the acceptor, the electron can pass through the tunnel mechanism, which means that it can actually work as a wire. However, when we use DNA and we intercalated a chromophore to absorb the light and trigger the electron transfer, we show that the efficiency of the transfer from the donor to acceptor, it decreases as the distance increase. So actually, we cannot use DNA, cannot be used as a local wire. So anyway, so that was a very cool stuff, and we used actually femtosecond spectroscopy. So the late Nobel laureate Ahmed Zuhail, he's one of the pioneer of this technology in the 80s. So I had to learn this and work on it, which was really cool at that time. Now that I have my poster, that's because of that. Now it's something, a personal story that I would like to share during this time. So I spent, I think, like 3 years at Northwestern, but 8 months of these 3 years, it was a decision that I wanted to visit Lebanon, and a lot of things happened from the time that I applied a visa for a student in 2000 or late 1990, 99, and 2007, I think. Without going into too much details, I decided to go make a visit. I had an offer in my hand for a job at actually Procter & Gamble, very well, big, huge company, global. But I, for some reason, and this is something that I live by, I believe that God has plans for us, whether we know what is the plan or not. I just believe that it happens for a reason. Anyway, I made a decision with my family to. I had my first newborn with me, and we visited the family in Lebanon, and went to the Safara, to the embassy, and they told me actually they need a security check, and I got stuck in Lebanon for 8 months. This is one of my most challenging part of my life. 8 months was very, very challenging, whether it's on personal or my career. As you can imagine, I had a job offer in my hand, so I was planning to go back and move into a company, start my career. Of course, all of this went went away, but what it really meant to me is that the support that I got from very specific people, mainly, and of course, Bilal. So Bilal, this is where my relationship started with him. He was one of the very few people who I really admired for what he did. He opened his lab for me, so I joined his lab for several months. But not only that, he facilitated for me to teach one course, organic, at LAU. So during this time, I was able to keep myself busy, get some income to some extent, because I wasn't working in the US. I had a lot of bills I have to pay. Just give you a flavor of what kind of people in your life makes a huge difference, and I'll be in debt for him for my rest of my life. So thank you, Bilal, for your generosity and for being who you are, and for friendship. So keep something for you guys, the young generation. Keep looking for these kind of individual in your life, and never lose contact with them, because this is very rare. Even though they are around all the time. There are good people all over. You just need to be able to be open to them. Anyway, moving on. So after 8 months, I get my visa. Went back, spent a few months in my postdoc, and I start applying for work. And this is where I interviewed for Alkermes. It's a company in the Boston area, so I had to move again from Chicago to the Boston area. Alkermes is was known in the early 80s, started in the early 80s, as a drug delivery company. And their platform was to use drugs that are already in the market. Think of any like Panadol or Tylenol, or any, of course, more complicated for different indications. But they have a platform where they embed this drug into a matrix, polymer matrix, making them like spheres in the micron size. They call it microspheres. They injected them into either the muscle or subcutaneous, and they form a depot. And this depot releases the drug slowly over time. And there was a lot of interest and need in the market. And this is where I start learning about why drugs are being developed. Different people develop for different ways, maybe making money, being famous. But I was lucky enough that at Alkermes, I learned something really that I carried till this day. Who we serve are the patient. We really need to think of what the patient needs and try to solve their problem. So in this case, when I joined, there was a program, or there was a molecule that Alkermes was interested in, and it's an antipsychotic. It's called aripiprazole. It's a drug that is in the market called Abilify to treat schizophrenia and bipolar disorder. Now, why long-acting injectable is a need for these antipsychotic? People who are schizophrenic typically take their drug from daily, an oral. Over time, they feel they're doing well. Like they feel very good, that the drug is working well. But for some reason, they decide, why do we need to keep taking all these pills and drugs? So they decide to stop the pill. Unfortunately, because schizophrenia is an actual disease, like cancer, like heart disease, like there's a relapse for every time they stop the medication. And every time they relapse, they get hospitalized. And there are a lot of research that the brain function, every time they get hospitalized, decreased by 50%. So imagine a patient who gets hospitalized twice, how much of their brain function goes down, and they be incapable of functioning in their life. So when you provide them with long-acting injectables, such as one injection every 1 week, 1 month, 2 months, or 3 months, they are not in control of taking that medication out. So for this reason, Alkermes did a lot of research, and they have several drugs in the market to address some of this a need for patient and for healthcare provider. So when I started on the bench, doing some synthesis of these new molecules. And the goal is to make a really low soluble molecule of aripiprazole. We took the approach of prodrugs. So a prodrug is a chemical entity that the drug has been modified chemically, like adding a functional group chemically to the molecule. Once it's administered, the body will metabolize it in a way to remove that functional group and provide the active after metabolism. The prodrug itself should be inactive. So it's naive. It's not doing any activity. It has to go through this metabolic pathway to provide that active drug. And a lot of research, in literature for many years, took the approach of prodrug for different reasons. Some of them to increase solubility, some of them to change the property. For us at Alkermes was mainly to decrease the solubility to make a depot once it's injected into the muscle. That first molecule, aripiprazole, again, I consider myself very fortunate and lucky that my first program at Alkermes ended up to be a product in the market. So it took, I want to say, maybe 8 years to go from the molecule that is shown here, this is all public information, to the picture that is above, and it's called Aristada and Aristada Initio. So this ended up to be a product in the market. People and patients are using it as we speak, and they've been using it for maybe 8 years now. Very proud of being part of this team. It takes a village to make this, but again, the journey and the amount of learning that I had to go through, it made me who I am right now. And I can give you a few examples of that. The first is we had to do what we call the human factor to develop this syringe that is shown here. And what it means is that as we're developing the formulation or the product, because the product is a syringe inside a syringe, how does the patient, or in this case, the healthcare provider, like the nurse, is going to interact with this syringe to administer to the patient? And you can only imagine how many mistakes could happen. Some of them could be grave, could be very critical, could lead to really grave results if it wasn't administered or handled the right way. So we had to meet with a lot of nurses, doctors, do a lot of interviews. I had to stand behind when in the movies where in the police department, you can stand behind a double mirror where you can see, but they can't see. We had to do that where someone is interviewing the doctor, interacting, and we're taking notes behind the scenes. So that was very cool. This helped a lot. And this is something that the FDA, the regulatory agency, expects you to do and to provide this information. Anyway, the other thing that I was really proud of is developing the Aristada Initio, which is what we call life cycle management. This is a product that came after Aristada, and to solve some problem that we identified in the Aristada. And the other cool thing is we were taking part of naming actually the product, like Aristada. How did this name came up? And why Aristada? Why not something else? So it's a long process that we had to go through at Alkermes. Again, I had the opportunity to learn about polymorphism. This is something that I learned on the job. It's a field that is well known in the pharmaceutical industry and in academia. I had a great team that we were able to publish a few papers while I was at Alkermes. And quickly at the end of my time at Alkermes, I had the opportunity also to work on biologics. So far, everything I talked to you about is what we call small molecule drug. But there's another aspect of the biopharmaceutics is developing biologics, which is protein therapeutic, very large molecules, and developing that is takes a slightly different path and different skill set, different needs. So lucky enough to be part of that, and I led this program. It's for oncology, for cancer treatment, different type of cancer treatment, and anyway, this went all the way to phase 3 after my time, but unfortunately, it didn't move past that. All right. After Alkermes, spent there, 12 and a half, 13 years at Alkermes, basically made my career at Alkermes. And I very fortunate to be part of that and really give all the credit to my mentors and my team there at Alkermes over the years. But at some point in the Boston area, there's a lot of biotech and small companies that keeps popping up, and really interested in knowing what's out there. So actually during COVID, where if you were following what was happening in the world, and especially in the biotech, not only in biotech, I think in other industry, they called it the Great Resignation. A lot of people resigned their post, whether they made drastic changes to their career or just tried something new. I guess I was part of this Great Resignation, so I made the decision to jump into a different company called Lindra. At that time, and it was a very small company, privately owned. But what intrigued me, a couple of things. First, the leadership or the hiring manager. He was the one who hired me at Alkermes. He left 5 years after me joining Alkermes, but I stayed in contact with him. He joined Lindra later on, and I reached out to him and I said, okay, well, would love to work with you again. So I joined that. But the other aspect of that is their technology. Now, I talked to you about long-acting injectables, where the patient will take one injection using a needle and a syringe, either into the muscle or subcutaneous, over 1 week, 1 month, 2 months, etc. What Lindra was doing is long-acting, but oral drug delivery. So as many of you, and as you know, a lot of people don't like to be sticked into needles, right? It's painful, it's invasive. So the easiest drug delivery path is definitely the oral route. So what Lindra was designing, or their platform, is taking a pill that goes into the stomach. The pill is, it's like these oil capsules, relatively large. But once it's in the stomach, it opens into a delivery system like a star shape. So Bilal, if you go to the next one, I have a picture here. And this star shape shown here with the hand, because it has that shape and that size, it stays in the stomach. It doesn't go through, it's not small enough to go through the pylorus and go into the lower GI. And as long as this is in the stomach, you can see it has different colors, and these are biocompatible polymeric material. Some of these that are colored, it has the drug. The drug will release in the stomach slowly over a period of 1 week, 1 month, or more. So this was really kind of a very novel idea. It came from Bob Langer's group in MIT. As you know, Bob Langer is a huge name in the biotech, founded many companies. One of them is Lindra. So fortunate enough to meet with him and attend several meetings with him on the board. But the goal for that is really to decrease or address the need of the patient. A lot of patients don't want to be on injectables, don't want to be sticked in their muscles or subcutaneous every month, even every month. So much easier if you take it once a week. And again, it's a long acting because they cannot stop every day. They have to remember only once a week, or once every 2 weeks, or once a month. As you can see, to build this drug, and this is only one pill, the engineering part of manufacturing that was really, really complex and very innovative. So you can only imagine what kind of challenges we had to go through, and the plot here showing actual clinical data. These are patients, and the concentration. The y-axis is the concentration of drug over time across the x-axis, and you can see this with the blue is how much concentration is in the blood over 7 days. And there might be like a, if you can see the like a dotted line. As long as you're above that dotted line, you have efficacious activity. So the product is working. So when I joined, we were about to move into phase 3, which is the last phase in the clinical trial for drug development. And there's a lot of work need to be done. At that time, I was leading the analytical development, which means to support all the analytical testing, developing the analytical methods, releasing batches to the clinic, putting it on stability. And I have a slide after that just to give you a flavor of what drug development is. Anyway, so 2 years and a half at Lindra. It's a small company like any other small companies, privately owned. Funding could be a challenge for small companies, depending on the stage. So we started, burning a lot of cash. We start, unfortunately, laying off some people over the year, and after 3 layoffs, I was impacted at the end, the third one, and I reach out to my network. Someone that I know from Alkermes. Actually, he was the chief medical officer at Alkermes, and now what is a chief medical officer at LifeMine. He, responded to my message and said, "Listen, we'd like to have you in, go through the interview." And fortunate enough they created a role for me. Because they wanted someone with experience of long-acting injectable, Alkermes work, for what they wanted to do. And as Bilal mentioned in his introduction, what LifeMine is doing is discovering new molecule from fungi. And when I joined, it was we nominated our first molecule ever for the whole company, worked on designing and delivering the formulation. Currently, we are in phase 1, so the drug is being taken by human. We're expecting some data toward the end of the year, and this is it's been really an exciting journey so far, and I'm really looking forward to what's next, where we can help and deliver these important medicine for patient. Just a quick, what indication we're interested in for LifeMine for the drug that I'm working on. It's for organ transplant. So people who get a new organ, whether it's kidney or heart or liver, they have to take medicine to decrease their immune system so they can accept the new organ. And unfortunately, there's only one drug in the market approved that is very efficacious, and a lot of patients take it. However, it has a lot of side effect, and so we're developing a new drug, hopefully that will be a replacement with less side effect, and maybe taking it less frequently for the ease of patient. Anyway, as I said, I tend to talk a lot. Bilal, how are we on the time? All right. Can you go back one slide? This is what takes drug development. It's a triad. There's the CMC, which stands for chemistry, manufacturing, and control, and this is where I live. Mainly chemistry, CMC. There's the non-clinical, which is all the animal testing and in vitro, in vivo testing. And then there are clinical, which is the human testing. And to develop a drug, we have to work on all of these three and work together to deliver a drug. Next slide, Bilal, please. And CMC, as I mentioned, is chemistry, manufacturing, and control. I just have a couple of, if you go into the animation, just chemistry is basically making the drug, testing it, making sure that it's stable, it's safe, and then manufacturing is how to make it in large amount to scale up. We can do it maybe in the lab on a small scale, but to make it commercial, you have to provide the need for patient. And the control is basically what's the quality or whether it's quality assurance or quality control that oversees these activities, specifically on the manufacturing and GMP batches, which stand for good manufacturing practice. And it's a highly what do you call it? Anyway, it's over the oversight is from regulators, highly regulated industry. We have to abide by regulations and interact with regulatory all the time, whether it's within the US or OUS. So it takes a lot to develop a drug, and there's no surprise that you hear like a new drug can take up to 10 or 15 years to make it into the market. Anyway, I'll stop here, Bilal, and give it to you. This is kind of a pivot from science to leadership. I put this slide. Maybe this can be a good segue for any questions that the team might have. This is a model that I learned from my former coach and my current friend Matt Marisola. It's called the bike model. If everyone is aware of the bicycle, there's the rear wheel and the front wheel. The rear wheel is basically what advances you, right? You bike, and this is what advances. However, to guide you where you want to go, it's the front wheel. So typically, as scientists, as a young individual, you really focus on the rear wheel. It's your technical skills, something that you learn on the job, or something that helps you on doing your job. However, to guide you where you want to end up, you really have at some point, and I would say as soon as possible, and maybe at even as you're growing your rear wheel, you really have to focus on your front wheel, which is more of the interpersonal, the emotional intelligence, the communication, the teamwork, how to build trust, how to build your network. All of this is in the or what people refer to historically soft skills versus the hard skills. Anyway, this is something that I really enjoy as a model, and I really share it with my mentees and people who I interact with. Anyway, I'll stop here. Sorry if I took more than.
Thank you so much, Tarek, for this beautiful narrative of your story, which is very inspiring on so many levels. I remember very well when you got stuck here for 8 months and in the middle of your postdoc at Northwestern, and it was an interesting time, but you ended up working in my lab, training my students, and we got a paper out of it. I believe it was in tetrahedron back then. It was beautiful to have you in the group back then. I was happy when you got back your visa. Not that I wanted you to get stuck here forever. We had that conversation before. So for the attendees, please enter your questions in the Q&A box. Say who you are, what's your major. I will be reading the questions, and I'll give Dr. Zeidan the chance to answer. But before I go to the questions from the attendees, I will start with the Crystal. Your question?
Thank you very much. And building on what you tackled regarding drugs and cancer, can you detail some of the challenges you faced while designing the drugs?
A lot of it's all about challenges. The interesting thing of a drug is something new. It's you're going into discovering something new, and I think it's a mindset. You have to be on a mindset of you're gonna fail, you're gonna find something unexpected, and going back to how to approach a problem. Grad school PhD is not learning something new. Yes, you learn something, but it's all about practice and training on how to solve problems. This is my approach of grad school. If not to publish only, or it's learning a new skill set. So for discovering or developing, there's all kind of challenges, from making sure that it is safe first to be given to the patient. There's a lot of controls that you have to put in testing, developing the method of testing, making sure that the method is giving you what you think it is giving you. So, it's all kind of challenges that you can think of in drug development.
Thank you.
Thank you. Farah?
Thank you, Doctor. Thank you, Dr. Tarek, for the inspirational talk. It was really amazing to get to know all your accomplishments. I only have one question. When you think about your legacy in the pharmaceuticals industry, do you view it more with regards to the patents you have filed, the medicines you contributed to bringing into the market, or the people you have mentored?
Thank you for this question. I think I would love my legacy is the people that I worked with, and the impact that I left with them, and what I taught them, and what we learned together. I can give you an example. The U.S. is huge. it's a continent. Boston is huge, but I have a close friend, a family friend, who works in Sony, and Sony has a department for medical devices, and out of nowhere he'd send me a text that with a picture with someone that he was visiting his lab, and this person, I worked with him at Alchemy's. We worked together, and the text of my friend saying, kind of, this person talked to you highly about you, and he misses working with you, and glad that I know you because you have a good reputation. And that's what I love to be my legacy, is what I left with, working with people, helping them growing in their career. I hope I did that. I hope I keep doing this. Yes, patents, drug development is great. If I have to put a second one, is basically the impact that I left on developing a drug. So again, it's a blessing, and I'm fortunate to be part of something that ended up in the market that patients are using. And one story that is very, very powerful for me, and I will never forget this. At Alchemy, we used to do these a town hall meeting with everyone, all like 600, 800 people come together in one place, and it's a full day meeting after meeting, going through a lot of companies do that. But we tend to bring someone outside the company. And in this instance, we brought a mother of a patient. Her son was a schizophrenic patient, and she came and presented, gave a talk to us, to Alchemy's. And I was fortunate enough that earlier that day, a small group of us, either the co-inventor of the technology or people who led the product during development, maybe a handful of people had breakfast with her. So she knew me personally, like we introduced ourselves and we had breakfast. So after she gave a talk to the whole company, she had to leave because Alchemist has to continue the program. So she left the room. After maybe half an hour, I left to go to the restroom, and I just bumped into her outside. And she just looked at me and just gave me a hug. And she told me, "Thank you for what you're doing. You really, had an impact on my son's ability to make a life. He's he used to be in the hospital. Now he has a career. This, even right now, I can be a little bit emotional, because until this time it was just a career, just a job. After that, it was a significant shift of, wow, what we're doing can actually impact human life. And this is the second thing. I mean, if both of them are equally something that I would love to be remembered by.
Thank you, Tarek. I should say that Farah is a chemistry senior student and Cristal is biology senior student. Now, the next question is coming from Leticia Nassar, biology student. The question is, the pharmaceutical industry operates across borders. How has working with international teams or markets influenced your approach to innovation and leadership?
Yeah, so I talked about the Erstada Initio, which is the second product that went after Erstada. And, I led the team across 3 different sites, one in Boston, one in Ireland. So we had a manufacturing site in Ireland, and the third one is our manufacturing in Ohio, in the US. It comes with some challenges. First, time difference, then culture. I love the Irish culture. They're really, really amazing people, very well-hearted people. I get to know them very well. But for me, the main thing, and this is, I remembered vividly, as I was responsible as a CMC lead to develop this product, the first presentation as a CMC lead, my main goal was to align everyone to be on the same page. And the most critical thing was communication. So I had like communication 3 times. This is the most important thing. If you can have an open communication, very effective communication, regardless whether you're 5 hours difference or 12 hours difference, you have different culture norm, as much as you can focus on communicating, having an open communication, which build trust. And as you build trust, any issues or any challenge that you can face, you can solve it as a team. So this is my approach, making sure that the communication is transparent, is open, is very effective. And over time, you really have to build credibility and trust among the team members. And all problems will take care of themselves.
Thank you. This question is coming from Carl Sawaya, chemistry student. What role have mentors played in your career, and what advice would you give students on finding and nurturing mentor relationships?
Oh, yeah. It played a very, very critical. I strongly advise you to look for mentors. Mentors will come along. You just need to be aware of them. And you might have multiple mentors. You don't have to have one mentor. Bilal, Dr. Kaafarani is my mentor. I reach out to him sometimes for advice. And this is the role of mentor. It's not to be next to you. There's a difference between coach and a mentor. A coach is a certain, period of time, typically, for a fee, teaching you something new. Mentors are people who you rely on for advice and for suggestion. I was fortunate enough to have multiple mentors and still have them. I still reach to them when I need. And then there are a third kind, especially for your career, that I really suggest that you look for, is a sponsor, what is called a sponsor. A sponsor is a person that will be representing you in rooms that you're not present, whether it's on the C-suite, on the board, on leadership meeting. You're not there, but they are there for you. So these are the 3 kinds that I was fortunate enough to have met some of the people. Mentors could be your sponsor or vice versa, but definitely mentorship is a very powerful resource that I strongly advise everyone to have and build. And mentorship is not a one time in time or space. It's a continuous, and you have to nourish it, and you have to keep. And it's a two-way. You have to give to get. It's not only one way. Hey, Bilal, Dr. Kaafarani, I need a, and then doesn't hear from me for like 5 years or no. No, you have to keep building that relationship, so you can get something in return as when you need it.
Thank you. This question is from Ali Dahir, biology junior student. What would you tell someone who is having trouble connecting what they learn in school with real life biotech uses?
So, if I understood the question is, I learned something in school that I'm not seeing how I can apply it in real life, or is there a disconnect between, like, I learned something and I learned it that it's the wrong thing? So I didn't understand the question well.
He's basically asking how you are able to, whatever they learn at school, if they want to take it into biotech, how do you make this connection? What is the challenge basically to use the knowledge that you acquire in university to go into industry and biotech?
Yeah, I mean, I can give my own experience specifically on the grad school to making the pivot to the industry. As I mentioned, it's not the substance, it's not like the endiene or the photochemistry that I learned and published. Is the training, is how I approach the problem, regardless of what's the problem. When you look at, I got this data, the question is, first, did I expect this data? If I didn't, why it didn't expect it? What went wrong? Is it something that I did in the lab? Is it something that I did during the analysis? It is something that I didn't expected. So if it is not expected, go back into research in the papers or in the whatever has been published, and see if I missed something. So it's the training that helped me make this pivot, not actually what I did in grad school or at school. And one thing is about learning: never stop learning, never ever stop learning. Live to learn. For me, I keep telling my folks is the day that you stop learning is the day we're dead. So as long as you're alive, you're breathing, learn something new. Make it either intentional or unintentional. Be a learner in your life. Learn something new. Everything that I published at Alchemies, I learned it on the job. Has nothing to do with my grad or postdoc. It's something that I learned on the job. Something that I do right now is what I learned on the job.
Thank you. This question is from Jean-Paul Castoun, biology student. Considering that in 2026 it's going to be the 160th anniversary of AUB, what was your fondest memory as a student at this honorable institution? Getting stuck in Beirut, no?
Well, luckily it was after I graduated from let's see, from AUB. This is what I shared, really, that my getting to know someone who I highly respect, Professor Makhluf Haddadin, the late professor, may he rest in peace, and his giving me this opportunity to work in the lab, and with the very, very rudimentary, very limited resources compared to when I joined, when I moved to the US with the cutting edge lab equipment. I remember in his lab, TLC, thin layer chromatography. Right now, when I joined, when I was in grad school, we just push a button and we order thousands of them. At AUB, we had to make them ourselves. Put some silica in dichloromethane, put the double edge slide, microscope slide, dip it several times. We had to make these things here, but as challenging these days. It was during the summer that I worked. It was very hot in the lab. The smells was very strong. this is what made me share these stories right now. So it made me who I am. So if something that I can remember from AUB is this summer that I worked in the lab with the late Professor Haddadi.
Thank you. This question is from Taleen Sariyuddin, chemical engineering student. As a person who has evolved from being a BS chemistry graduate to a VP of pharmaceutical sciences at LifeMine, what are some advice you might offer to an early career seeking individual striving for success?
Be yourself. Don't be someone else. If you like chemistry, then do chemistry. If you like biology, do. If you like medical, do whatever you like. Once you enjoy what you're doing, things will fall into places. Be open to learn, as I mentioned. Be a learner. Be humble. Like someone could be 1 year more experienced in the industry. You might think them less of a smart than you. Don't. You might learn a lot from them. As you grow in your career, whether in leadership, or always think of, you can learn something, at least one thing, from other people, whoever they are, whether they're people that you like or you don't like. Even the people that you don't like, you can learn something from them. They can give you the experience of how to deal with difficult people, how to behave in front of them. Take it as an opportunity. Don't run away. Don't say, oh, I'm not gonna. No, push yourself to learn something new and read, educate yourself. This is something that I strongly advise. I'm happy to share offline if you'd like. Feel free to stay in contact with me. I'm on LinkedIn. I can suggest books that you can read about leadership, about emotional intelligence, about mentorship, etc. This is something that a topic that is really dear to my heart, and I keep educating myself and reading about it.
Thank you. This question is from Amina Samad. What advice can you give that you think would be the most honest advice about what you have been through?
What is? Can you repeat the question? Sorry.
What advice can you give that you think would be the most honest advice about what you have been through? Not coming to Lebanon for 7 and getting stuck in Lebanon.
Life will throw at you different kind of challenges. To be really honest, have faith in God. I mean, this is for me. I believe there's one God, and I believe in Him. Whatever happens for a reason, and there are things that I, as many of you had to go through. We might not know how it will end or at that time, but I always had faith that it will work one way or another for the better for me. So that's my most honest answer for that, regardless of the challenge that is. I'll give you an example. When I was graduating from Florida State, unfortunately, I wanted to join a professor at MIT that we met at Florida State, and for some reason, my advisor didn't want me to join for one reason. This was very stressful, very, very kind of a shock for me. But I ended up joining Northwestern University, get to meet Fred Lewis, who was very, very supportive, especially when I was stuck in Lebanon. So it worked out well. And I can tell you, okay, this is something I can tell you. That same professor at MIT, there was a grad student who graduated from Northwestern, who I collaborated with and we published a paper together. He graduated while I was a postdoc, and he joined this lab, the lab that I wanted to join at MIT. That is like 2 years later. I stayed in contact with him. He was miserable in his lab. And the reason is that the lab was very competitive. People stab each other in the back. The professor is not available for his student. He has a huge... So now thinking about it, that's 2 years later or 3 years, thinking, oh, I should have gone to MIT. Why he didn't let me go to MIT? Now I think, thank God that I didn't go there.
This question is coming from Gael Hankash, who's biology student. Gael has been tweeting live about this talk, as well as she's posting on LinkedIn. She tried to tag you, but she couldn't. I told her because you're not connected. So once you're connected, then she will tag you and they will appear on your page. She is WhatsApping me as we are talking. What's the biggest scientific risk you have taken, and how has that shaped your career and way of thinking?
Good question. The biggest risk, scientifically, right?
Yes, we are only talking scientifically, Tarek, not personally.
You know, I would say developing drug, especially when you're in the leadership, you have to take risks. You know, when you're responsible of a function such as chemistry, manufacturing, and control, and you're the person who's going to answer to first to the leadership of the company, to the board, to people who are investing in the company, but more importantly, who has to answer to the regulators, or God forbid something happen in the clinic, you know, either an infection that the patient had that could be triggered back into the drug product, or you know, we call it drug product, or the clinical trial material that the CMC lead or the head of CMC is responsible. That's always a risk. So as you're developing those material that's going to end up being tested in actual human, you really wanted to make sure that it's safe, that is, and there, you know, it's definitely you can do that, but now add to it timeline and push from, you know, investors and you know, leadership saying, listen, we want this material by the end of the year. And, you know, for us, if we want to make sure that everything is in plan and everything, we need an extra 2 months. No, you're not going to get 2 months. So what can you do? You're going to have to take some risk. You're not going to have all the answers in front of you to say, yes, we're ready to go into the clinic. You only have 50% of the answers, and then the other 50 you have to take a risk. So it's always taking risk in drug development, especially in order to push it and answer, you know, to the investors, to the market, to the leadership. So hopefully I asked.
Can we stay 10 more minutes, Tarek?
Yeah, I have time.
Okay, thank you. This question is from Jean-Marc Rebez. He's also a biology student. While studying at AUB, did you ever think to follow the pre-medical track?
No, never. Never. I from the beginning, I want to do just chemistry. I love the material. I think after 1 year of chemistry, I really wanted to do research. And this is where I worked in the lab. But I wasn't planning to take pre-med courses. The electives was all, you know, I had a business and I had like, I think biology 210 or something that is not medical pre-med. It's just a choice that I wanted. I really like research. This doesn't mean that if you do medical, a lot of medical students do research. Like it's amazing. We work with medical people all the time in research in biotech, and very, very important function, significant important. So it's just me personally, I just wanted to do, you know, basic research for chemistry.
Thank you. This question is coming from Antonio Alhaj, PhD student PhD student here in biomedical engineering at AUB. Thank you, Dr. Zeidan, for sharing your journey and for being a beacon of hope. I am a pharmacist by education and had 3 years of experience in industrial pharmacy, particularly in product development of OSDs. I'm curious to learn more about the mechanism of drug release for links and the reasoning behind its star-shaped design. Do you think establishing a local startup for R&D of novel DDS would be a viable direction in light of regulatory complexities?
I'll answer the last question first. Yes, it's always worth it to pursue what you believe in. Make sure that you do your homework. Don't think only of obstacles. Like if you have an idea, it's easy that the brain will tell you, oh, you have to deal with it. Oh, if you have enough conviction on what you believe, go for it. And make sure you have a good support system, whether it's mentors or investors or whatnot, and keep learning. If you have small successes, doesn't mean that you know everything. I bring it back to the learner behavior or mindset. So that's probably the question, the last one about the mechanism. So there are different ways of delivering long-acting injectable, and I'll get into the oral delivery. For injectables, there are different technologies. It started earlier in the 60s where they dissolved something into oil. Oil is hydrophobic, so it acts as a depot once it's injected either into the muscle. Haldol is one of the oldest drug in the market for antipsychotic. That is a monthly injectable. But then it comes with disadvantage, like it's painful. You have oil in the muscle. It is painful. Then it comes the microsphere, which is biodegradable polymer, PLGA, matrices putting into the drug. The mechanism of release is governed by the hydrolysis of the polymer in the body, in the injection site. For something like Aristada, the work that we worked on at Alkermes is mainly governed by the solubility and the dissolution rate of the crystals of the drug. So there's no polymer there. It's just pure crystal drug in the depot. And it depends on how fast it dissolves. Particle size, smaller particle size will dissolve faster, larger particle size will dissolve slower. And there are other technology out there, like in situ gel or other. Anyway, as for Lindra, the reason it stays in the stomach is because of the size and the shape. So it was designed from the MIT lab in a way that it has to tolerate all the mechanics of the stomach during food, during fasting. All of this, it has to stay the same shape and is flexible enough, but it's big enough not to go through pylorus, which is the lower exit of the stomach. And but it is also designed by biodegradable polymer that you can design it in a way that this polymer will degrade in the stomach pH over a certain period. So once those polymer in the arms of the star start falling apart, those arms will break. So you have a smaller shape, and this will enable going through the lower tract, and it exit by the GI as naturally. So that was the idea of that. Unfortunately, this company closed earlier this year, but the idea is very intriguing, and I hope at some point whoever bought that platform can make it to the market.
Thank you. This question is from Leah Slim, Environmental Health. The drug development process is full of uncertainties. How do you stay confident when faced with long timelines and unpredictable outcomes?
Yeah, it's it's the nature of our industry. It's discovering new things, a lot of uncertainty, not only whether the drug is going to work, it's the uncertainty of the economy, there's the uncertainty of the market. My big advice for you guys is there's a model that I call it circle of control. So imagine a circle, this is circle of control. You're in control here. Now behind this is a bigger circle. It's called the circle of influence, right? And then outside this circle is the circle that you're not in control of. If you spend time in the circle of out of control, you're paralyzed. You can't function. I'll give you an example. You plan a barbecue for Saturday. You had everyone invited, and you spend a lot of money, and then it, start raining. Forecast is saying, oh, it's going to be very cold. And are you in control of that? No, you can spend why is this happening? And spend your energy worrying about something that you're not in control of. My advice is stay in control, build the circle of influence, and this is by building your network, building your credibility, so you can be able to influence decisions. And this is where sometimes you want really to spend some time. But during tough times, stay in the control, make sure that you're sane, you can do it. If we need, every time we need to start a project or start to make a drug and worry about, oh, is it gonna be a success or not? We're not gonna be able to move. So at this time, what can you do? I can, maybe raise some money to start this idea in the lab. I need a lab. I can go and find a lab, work on that. This is what's in your control. Whether this is going to end up in the market or not, or successful, doesn't matter. So that's my, hopefully this answers the questions of the answer.
Thanks. We still have 6 minutes. Let me try to get more questions. Marianne Roche, medical lab student. If you could go back and give your AUB self one piece of guidance about navigating uncertainty in science and career, what would it be? Philosophical, little philosophical.
Wow, yeah. I mean, this is amazing. I'm this is really refreshing. It's like a breath of fresh air of having, getting to hear this young generation's questions. I'm very, very, very impressed. And thank you for challenging me with these questions. Maybe it's a cliche answer, but I don't like to look back and regret. I always look back to learn, and because it's back to my philosophy of life. Things happens for a reason. I don't have control of it. I believe in God, and God has planned for me, whatever, and it's the best thing for me. Always happen, whether I see it right now or not. So if I were to answer it in a way that what I could go back and learn rather than regret. If I got that question right, because she mentioned regret, what I would do at AUB is be a little bit more social. I wasn't very social. I'm an extreme introvert, if you don't notice, so I was a little bit of my wife keeps telling me I'm an antisocial person, but I would be more social and building more relationship and network if I were to go back. But it's not a regret because it made me who I am. So.
This question is from Hanna Basil, biology senior student. In your journey, what habits or mindsets have helped you stay resilient through setbacks, and how can students start building those today?
Yeah, it's as I mentioned, having a learner mindset, and really build strong relationships, build these relationships on trust, making sure that you build trust with individuals that you care about and they care about you, whether personally or career-wise. So it's something that really it's a journey. Take it as a journey. I had to go through an exercise during my career, especially at Alchemist toward the end. I was and as a senior director at that time, and again the support system that I had was amazing. So I requested a coach, executive coach, and that coach, Matt Mirasola, that I mentioned, the bike model. I worked with him over a year and a half to articulate my values. And I really urge you guys over time. Maybe you're still young to have, but maybe it's never late. But once you articulate your life values, which should be the same as your career, and live by your values, all challenges, everything will you can achieve anything, and you'll be happy. Life is, you really need to be happy in your life, and it starts with doing what you like, identifying your values, and make decisions based on these values. Live by them. So hopefully that answers the questions. And again, please feel free to reach out anytime after that. I'm happy to stay in contact.
You're gonna get a lot of requests for on LinkedIn to connect. Two last questions.
Sure.
This question is from Jad Kanaan. As co-inventor of LinkRx, how did the idea come to life, and what were some of the biggest scientific and logistical challenges you faced during its development?
Yeah. So the LinkRx, the name actually comes from linker. So not to go into too much details, but as I mentioned, there's the active, and we want to make a drug. But this active, if you look at the structure, it doesn't have a lot of functional group that you can attach to it. And whatever the functional group, which is the amide functional group, is very stable. So if you attach anything to it, it's not going to be metabolized. So we had to add a linker in between to make it, to add whatever we want at the end. So this is the linker X. So RX is prescription in the pharmacy. So they played on linker and prescription. So most of the challenging is basically, if I can remember from a chemical perspective, because of this linker, once it's metabolized, it's a two-step. It's removing that function that we added, this very lipophilic tail, and then the linker to give you that active. But sometimes this linker stay attached. And for regulators, this is a new chemical entity. So they are worried that it's a new drug. So we had to develop data showing that once this is hydrolyzed, the lipophilic tail is hydrolyzed, this linker had a very short lifetime in circulation. So it's not built up in systematically that could lead to some side effect. And it's chemically driven, it's just water mediated hydrolysis or rearrangement to give you the active. So I think this is one of the things, because this is new, that nobody did this before. And we had to build that, of course, as a team, as an organization, from non-clinical and chemistry, and etc. So hopefully that answered the question as well.
Last question, though there are many more questions, but for the sake of time, this question is from Leah Ramal. With AI on the rise in most scientific fields, including designing molecules, do you see it as a collaborator, a competitor, or just another tool?
It's definitely a very strong tool. And going on the learner, this is something that I'm learning right now. I'm intrigued, as probably many of you, to see the bridging this making this new bridge between drug development, whether it's discovering new molecules, new drugs, or actually formulating the drug, or streamlining the development of the drug, the scale up, and how does AI can help us to make it safer, less risky, because it's all about decreasing the risks, and more effectively. So no, it's not an enemy. AI is a great tool, is a very powerful tool. We're just scratching the surface of it. So I don't know how we're going to use it. I can tell you there's a lot of discovery. Actually, LifeMine uses AI and machine learning to discover molecules from fungi. So this is where it started. It started, in discovery, and how it's going to be on selecting the right excipients, the interaction between the excipients and the API or whatever. This is still being developed, and there's a lot of effort going there in the industry on that.
Well, unfortunately, we have to stop right now, but thank you so much for this beautiful and inspiring story. And I'm sure many of the attendees have many more questions that they may reach out to you on LinkedIn. I have a sensor here. When I don't move that much, the lights go off. So when I am waving, I'm just waving to the sensor. I want to thank you again, Dr. Zeidan. And as I said at the beginning of my opening remarks, welcome home. This is your home. It will always be your home. And I look forward to seeing you here at AUB. I might see you earlier, though. You're still in Boston now, right?
Yep.
Yeah. So I might be stopping in Boston in March. Then I will reach out to you, and then we can reconnect. Our third mentoring talk is going to be on October 20th by another chemistry graduate, but this time he's going to be going into the medical field, and he will tell us his story. We will be announcing it very soon. Thank you again, Tarek.
Well, thank you, Dr. Kaafarani. It's been humbling and very refreshing, as I mentioned. Getting to listen to the questions, and please feel free anyone to reach out. It's been really great pleasure, and thank you very much, Dr. Kaafarani, for giving me this opportunity. I just want to end it on a note that I left AUB 25 years ago. What I see right now, it's a huge evolution, and I can, as you know, I just want to give a lot of credit to Dr. Kaafarani, because I've been following him from the TrEd career to the carnival, the chemistry carnival, to the Makhlouf Haddadin series, and bringing all these Nobel laureate and influencer to have it as a resource for the new generation. This is something very special, and I'm really proud of what Professor Kaafarani and his team, former and current, have been doing and achieved so far. I just wish you all the best. Continue to with the success. And if there's anything I can help with, let me know. It's something that I'm really proud of, to be associated and know Professor Kaafarani and being part of this series. So thank you very much, everyone. And till the next time. And hopefully I'll see you soon, Bilal, when you're in town.
I hope so. Thank you for very humbled with your beautiful remarks. Thank you so much. And thank you, Farah and Cristal, for joining us on this panel. Thank you, everybody, for joining and staying that late until October 20th for our third TrEd Career Chat. So long. Have a great night. Thank you. Bye.
Thank you.